A sleeping dog breathes steadily, completely at rest. To the naked eye, this physical stillness feels close to the absence of life. We instinctively use visible, external action to identify living organisms. However, macro movement is a secondary effect that fails to capture the complexity of biological reality. This microscopic cross-section visualizes the true biological baseline. Beneath the surface of every organism, there is a chaotic, continuous shifting and transport of molecules across cellular boundaries. These internal chemical reactions are relentless. Molecules constantly form and break bonds to maintain the organism's physical structure, operating 24 hours a day, regardless of whether the larger body is running or sleeping. The requirement for internal motion is universal. Viruses, for example, show no independent molecular movement when outside a host cell. That mechanical silence is why scientists debate whether they can be classified as living entities. The active maintenance of internal molecular movement identifies a living organism, even in the absence of external action. Maintaining that constant internal motion means constantly fighting structural decay. Doing so requires an uninterrupted input of external energy. Organisms manage this requirement through life processes, automated maintenance operations running in the background to keep the body's hardware intact and functional. These operations rely on four major interlocking systems: nutrition, respiration, transportation, and excretion. Nutrition is the foundational step. Before an organism can transport materials or execute respiration, it must acquire the chemical fuel to run those systems. Every biological system on the planet must secure this energy. The method they use to acquire it divides the natural world into two primary architectural frameworks: autotrophic and heterotrophic. The autotrophic framework is an exercise in biological manufacturing. These organisms synthesize complex organic food structures by harvesting inorganic substances directly from the environment. This equation shows that exact synthesis engine at work. A plant pulls in carbon dioxide and water and uses the energy from a photon of sunlight to force those molecules together, yielding glucose and oxygen. Executing this chemical conversion requires a specialized mechanical interface on the plant's exterior surface to physically capture those invisible atmospheric gases. In this diagram, stomatal pores act as regulatory valves controlled by guard cells. Through water pressure, they swell to open the pore, allowing carbon dioxide to ingress. Simultaneously, internal chloroplasts absorb light energy to drive the assembly. Plants operate as self-contained synthesis engines, locking free-floating atmospheric gases into solid, usable energy structures. How can we empirically prove that invisible carbon dioxide is a non-negotiable raw material for this synthesis? This illustration details a controlled isolation method. Two identical plants are sealed inside bell jars and exposed to identical sunlight. We introduce a disruption variable on the left, potassium hydroxide. Its function is to absorb all available carbon dioxide within that sealed jar. The experimental result follows. The plant isolated with the potassium hydroxide fails to produce starch. Cut off from the gas, its manufacturing engine stalls entirely. Removing this single variable proves that carbon dioxide is the requisite foundation for autotrophic energy synthesis. Conversely, the heterotrophic framework relies on extraction. Organisms that cannot synthesize their own glucose survive by forcibly extracting carbon compounds from other organisms. The most direct method is holozoic nutrition, a mechanical three-stage process of ingestion, digestion, and absorption. This process occurs even at the cellular scale. This sequence illustrates holozoic extraction by a single-celled amoeba. It deploys pseudopodia to encircle and trap an external food particle. The particle is sealed inside a temporary food vacuole. Here, chemical digestion breaks down the material, and simple nutrients are absorbed directly into the cytoplasm. Whether microbe or mammal, heterotrophs function as extraction units, dismantling external matter to survive. When we scale that extraction concept up to a multi-organ mammalian system, the mechanics become highly specialized. This diagram maps the human alimentary canal. Starting at the mouth, teeth mechanically crush material, while salivary amylase dismantles starches. It moves to the stomach, where hydrochloric acid and pepsin digest proteins. Finally, liver bile and pancreatic enzymes finish the teardown in the small intestine. This leads to the extraction climax. Millions of microscopic villi lining the small intestine absorb the newly freed molecules directly into the bloodstream. Digestion is a multi-stage chemical refinery, engineered to extract maximum available energy from the physical structures of other organisms. Both biological frameworks solve the same physical problem. The autotrophs synthesize energy, and the heterotrophs extract it. This split-screen animation contrasts the two methods side by side, a leaf constructing glucose from photons, and a human digestive system tearing matter apart to find that exact same molecule. Autotrophs construct the energetic baseline of the planet. Heterotrophs survive by consuming those structures. Regardless of the framework used to obtain energy, the ultimate destination remains the same. The mechanics of stomata, the sweeping arms of pseudopodia, and the acidic pipelines of alimentary canals all serve to fuel the internal chemical reactions happening inside our cells. Nutrition is the foundational life process, the acquisition of fuel that sustains the continuous molecular movement separating the living from the dead.